Okay, let's finalize and talk about our genetic and evolutionary foundations we're using in biochemistry. Now, when talking about life, it is important to bring up the hypotheses that we base most of this information on in the origins of life. Based on data that we have, the origins of life and formation of the Earth formed about 4.5 billion years ago. We know this mostly from things like carbon dating. And we know that life probably began between 3.5 to 3.9 billion years ago. This is at the simple cellular, unicellular level of life, but life nonetheless. Notice that this is a little bit more of a hazy date, 3.5 to 3.9 billion years ago, instead of, we know Earth was right around 4.5 billion. So actually limiting when life began is still a bit hazy. Now there are a couple hypotheses about how the origins of life came to Earth. And some of them that we will be talking about include the Oparin-Haldane hypothesis. This is a very familiar one. Many of you might have already heard of it. This suggests that life arose gradually from inorganic molecules. So building blocks that were already around on the Earth, like amino acids, etc., formed first and then started combining to make complex polymers, etc. And there's an experiment we'll talk about called the Miller-Yuri experiment, which provided evidence that these molecules needed for life could be formed from inorganic compounds, like suggested in the Oparin-Haldane hypothesis. Some scientists support what we call the RNA world hypothesis, which suggests that the first life was actually RNA that could self-replicate. And others favor the metabolism first hypothesis, which is that metabolic networks came before either DNA or RNA existed. There's also evidence that simple organic compounds might have come from meteorites to early Earth. But the main component here is that the formation of self-replicating molecules is a key step in the origins of life. The Miller-Yuri experiment, to expand on, was an experiment done by Stanley Miller and Harold Yuri. And again, they did this to test that hypothesis that Oparin and Haldane had. What they did is they found organic molecules that could be spontaneously produced under conditions that they assumed, the reducing conditions they assumed resembled those of early Earth. So they took this, this experiment in which they added molecules into, you can see this flask-like container, and they put it under the conditions of early Earth, so very reducing conditions with electricity present, which would have been present, and then found if they could produce some of these things like amino acids, and they showed that they could. Now, scientists think that the atmosphere of early Earth was a bit different than what Miller-Urey actually synthesized in their setup, so they don't think it was as reducing or rich in ammonia and methane as they assumed when they did this experiment. So it's doubtful that their experiment was actually accurate in its simulation, but there have been multiple experiments done in years since that do show that organic building blocks, especially things like amino acids, can form from inorganic precursors, and it's under a wide range of conditions. So there's evidence that these types of building blocks could have been formed naturally in early Earth. And as noted in relation to the availability of some of these molecules through meteors, recent teams have also discovered things like amino acids and even ribose and bioessential sugars, including xylose, in meteorites that are rich in carbon. Other building blocks have been found, and actually, I'll link this separately, but another even more recent article from NASA showed that even a very simple enzyme, very, very, very simple, it's only a few molecules long, but an actual enzyme with a site for catalysis was found in meteorites as well. So again, one of the possibilities is that the first life forms were just self-replicating nucleic acids like RNA or DNA, and that other elements like metabolic networks and utilizing small molecules were added on to this basic system. The reason for this hypothesis is that we know RNA can act to carry information. So we know it can carry genetic information. It can carry information, plans, blueprints to build things and make things. And it can act as a catalyst. So that's the important step. RNA can fold and act to catalyze reactions, and it can store information. This is one of the major points about RNA and the major pieces of evidence that suggests that it could have been one of the first self-replicating life forms. A possible scenario in which we started with RNA is shown here. Generally, again, we have the prebiotic formation of simple compounds. So like stated in that Miller-Yuri experiment and other experiments, we assume that building blocks like nucleotides and amino acids could have just formed naturally in the primitive atmosphere or in gases in undersea vents. And then production of short RNAs or RNA-like molecules from these nucleic acids that were produced could occur with random sequences. So natural polymerization of nucleic acids that were formed based on these simple compounds. This would then follow that we'd have selective replication, self-duplication of these RNA segments. We know that RNA can selectively replicate and self-duplicate. So RNA segments could then make more RNA segments. And as these RNA segments elongated and changed, there could be synthesis of specific peptides that was catalyzed by RNA. So now that you have bigger RNA pieces, remember, as I said, RNA can also act as a catalysis member. So it can bind to amino acids, and maybe the RNA started binding with those amino acids that were naturally formed and then starting to make peptides. So then we have this increasing role of peptides in RNA replication. So co-evolution could have occurred. We have self-replicating RNA catalyzing these proteins. Together, they're co-evolving. And eventually we get this primitive translation system. Translation system like what we see in cells now, in which RNA genomes and RNA protein catalysts exist. And then eventually the idea is that genomic RNA begins to be copied into DNA with the now addition of utilizing the deoxyribose sugars. Ultimately, we know in the end of the scenario whether this is the real scenario of beginning of self-replicating molecules to life, that we have a DNA genome translated on RNA protein complexes with RNA and protein catalysts. Genetic continuity in general is vested in these DNA molecules. This is a main genetic concept that you should know, and that, again, I'll be reliant on talking about in this class. So again, one of the most remarkable properties of living cells is their ability to reproduce. And they can do this for countless generations and nearly perfectly. And they do this through utilizing DNA molecules. DNA is a linear polymer made up of small polymer, remember, small units linking together, and it's covalently bound. So the units are covalently bound and joined by different deoxyribonucleotides. I brought these up briefly before, but remember those are the A, G, C, and T nucleotides, which you should know. And another concept is that DNA, of course, can replicate. Here we're showing a DNA replicating. It's showing two blue strands which separate into two new strands. And the two new strands, which are shown in pink, are synthesized, each with a segment that's complementary to the blue strand that is being pulled apart. Ultimately, it results in two new molecules with identical to the original, with one half from the original and one half newly synthesized. Now, again, this is a concept you should have already gone over in basic biology or cell biology, but if you're having trouble reviewing kind of the steps of DNA synthesis and what occurs during this process, I again have linked a video to go over to review structures of DNA and the components of replication. Other points to remember, of course, DNA is very large. It's usually coiled into such a very tightly packed area, but its molecular weight, which is based just solely on the amount of atoms there, right, is extremely large in the millions or billions. But despite the enormous size, the sequence of its nucleotides is very precise. And the maintenance of this precise sequence over a long time is how we have genetic continuity. That's how we have organisms that sustain as a species over time. The ability of DNA to maintain its precise sequence. The linear sequences of amino acids, which we'll be talking more about in the creation of proteins, should also know is encoded in DNA in the gene for that protein. So remember, what we call genes is a segment of the DNA strand that encodes for a protein. It produces the protein's unique three-dimensional structure, and that's a process that's also dependent on environmental conditions. Again, we'll be getting more into proteins and their three-dimensional structure throughout the semester, but understand where proteins are coming from. They are synthesized based on code called a gene within DNA. The central dogma of evolution and the central idea in biochemistry that we will be going over a lot, and we'll be talking about each of these parts, is this DNA to RNA to protein. Now, sometimes the central dogma, and I'll tell you this now, is not as linear as it is shown here. Sometimes we can just go RNA to protein or go backwards from RNA to DNA. And those are circumstances in which we say the central dogma is broken, or that a molecule doesn't exactly follow the linear central dogma. And that's okay, and we'll talk about those situations. But generally, you should understand this linear process because it is the kind of main process we see in which you are taking DNA and you're doing transcription of DNA into RNA. So this transcription makes something called messenger RNA. Messenger RNA then is going to translate the RNA into a polypeptide. This is taking RNA and going to amino acids and a protein in the end product. So again, DNA transcription to RNA. Again, this is specifically called messenger RNA. Messenger RNA is translated in ribosomes in the organelles of those cells to make then a polypeptide chain. This is just an unfolded chain. And then the chain is then folded into its final protein structure and then is active. So we take this unfolded chain and then fold it, and this folding occurs through different means. Again, we'll talk more about this to make the three-dimensional protein, which is catalytically active. Now, in talking about genetics, we of course have to bring up mutations and its role in natural selection. Mutations occur in general more or less randomly in DNA, and they can also occur in RNA. Mutated polynucleotides can then be transcribed and they can be translated through the central dogma idea into molecular machinery like proteins. So if you have an error in your DNA and then you go through transcription and translation, you now have potentially something that is a new protein, new machinery. Mutations ultimately give organisms an advantage in a given environment. And in those environments where they give them an advantage, they are more likely to be propagated. So this idea about mutations is that though all mutations might not be advantageous, ones that are advantageous are more likely to be propagated and then continuously propagated until eventually maybe you have speciation occur. So let's look at an example of where you have a favorable mutation. Here's DNA, and you can see highlighted within the DNA is a gene, right? So this is a segment of the whole DNA chain that then is going to translate to a particular protein. This gene is for hexokinase. This is a protein, the hexokinase protein, which is used in metabolism. So if a rare mistake occurred in DNA replication that duplicated the hexokinase gene, so now in our DNA strand, we accidentally have a second portion of it that has this hexokinase gene. And then, okay, so now we make two hexokinases from our DNA. That's not so bad. It's not going to be something that kills an organism, and it might just continuously be propagated. But now let's say we have a second rare mutation that results in the mutation of the second hexokinase gene. So this hexokinase gene that was duplicated one time accidentally, not really changing anything, not doing any harm. It's been mutated, and that mutation changes its protein. Let's say that mutation changes the protein to now make a hexokinase that can bind galactose. So original hexokinase doesn't bind galactose, it actually deals with glucose. But now our mutant hexokinase can also now bind galactose. So we have original hexokinase still being made, still does its same thing, still can utilize glucose, but now we have a new hexokinase that binds galactose. This is advantageous. We have galactose around, and now we can utilize that galactose as an organism. And so that mutation of the second hexokinase is favorable, and it will be continuously propagated. The evolution of eukaryotes in general is also thought to be mediated through this type of evolutionary mutation through something called endosymbiosis. The hypothesis here is that ancestral eukaryotes did not have this aerobic metabolism. They could only do anaerobic metabolism. But bacteria themselves are known to be efficient at aerobic metabolism through the oxidation of fuel to CO2. Ancestral eukaryotes are thought to potentially have symbiotically absorbed a ancestral bacterial genome. And then this genome was then continuously propagated within the eukaryote. This formed what we now call mitochondria. Mitochondria does have its own separate small mitochondrial genome, which is why we think that it originally came from a bacteria. It's a circular genome, like in prokaryotic cells and bacteria as well. And so we think that these then became symbiotic with each other and are now produced in tandem. Similarly, plant cells could have been derived by a secondary process in which you now have already absorbed mitochondria and then absorbed and endosymbiotically formed with a cyanobacteria genome, something that utilizes light to produce energy. And after engulfing this cyanobacterium, it became endosymbiotic, multiplied, and then were genomically reliant on each other. This could have ultimately made a photosynthetic eukaryote, something like a plant cell. In general, we now have an enormous amount of information from genomes. And this is something that's becoming increasingly important in science and in medicine. Now that we have this treasury of information about cells and their sequence of the genome, because we have the complete genetic endowment or the complete genetic code of many organisms, we can determine a lot of information. So when we have the sequence of a genome and we know each gene is its assigned function, so we know the genome itself, the actual code of an organism, and we know its assigned function, people like molecular geneticists can then group genes according to their processes, right? So if we know the function of a gene and we know the string of code and all the genes that are present in an organism, we can map that together and figure out what genes on an organism do what. And this general knowledge of the complete genomic nucleotide of an organism gives us insight into evolution, but it also gives us lots of opportunities in human medicine. So something like genomics, a topic we'll be talking about, which is the general study of all of the genes within an organism compared to the same organism within that species. So how is a person with a particular disease and their genes different from another person, same species, and same ultimate genome, but they don't have a disease, how are those different? How are those genes acting differently? Because we have this information mapped out, we can do these comparisons. That's called genomics. This is a field that is increasingly being tied to personal medicine. So we will be talking about and understanding what the gene is and how it functions is imperative to understand how we're utilizing genomics. Okay, so to summarize, we have gone through in depth understanding of what defines a living organism. We've talked about structures and function of cell. Those are important things to remember and know those different structures and how they function, differences between prokaryotes and eukaryotes, differences between animal cells and plant cells, and those organelles. We will be talking about those organelles in more depth. So remember what those organelles are and what they do. We realized that the structure of biomolecules offer given them specific functions. So we talked about those functional groups that are important, functional groups that we've seen in organic chemistry, and some new functional groups you'll be seeing in biochemistry. And we've talked about those very special molecules as ABCs, right? Amino acids, the nucleic acids, and the lipids, and also sugars and the phosphate groups. These are base molecules, building blocks that we'll be talking about over and over again. And knowing those structures will be very important. We also talked about principles of bioenergetics. So main principles that, again, you've probably seen in chemistry that will be brought up again through this semester, especially involving the energy required and the energy diagram of a reaction. So if a reaction is spontaneous or non-spontaneous, how you can calculate that and how you can use chemical equilibrium to change a reaction. We also talked about catalysts and types of catalysts we see in biochemistry. Those are enzymes and what enzymes do to that energy profile of a reaction. And then ultimately, we also reviewed forces behind evolution. We talked about the general idea of life starting and those concepts of going from RNA to now self-replicating cells. Talked about central dogma that will be very reliant on using and talking about more throughout the semester, DNA to RNA to protein. And we also just talked about how mutations are viable and the idea of genes and mutations in evolution and the genome itself and how important understanding the genome and the function of genes with these proteins is important for both evolution, but also for medicine. Okay, guys, I hope that was a comprehensive review. I know this was a long chapter. Again, it's a very long review of many of the concepts we'll be bringing up. And if you do feel weak in any of these concepts, now is the time to review them as we then go into our next chapter.